Hello guys, welcome to my channel. So as you know, we are studying about the protein function. So in the last topic we studied about the immunoglobulins. So now we'll study about the protein interaction that is modulated by chemical energy. That is like actin, myosin and molecular motors. Right. So I'll just write down the protein names here. And molecular motors. All right. So as we know, organisms move, cells also moves, organelles and macromolecules within the cell, they also move. So the movement of this all arise from the activity of fascinating class of protein-based molecular motors. So and that is fueled by the chemical energy used usually derived from the ATP, and the large aggregates of these motor proteins undergo cyclic conformation changes that accumulate into a unified directional force. That's a tiny force that pull apart the chromosome in the dividing cell, or then immense forces that levers a pound punching quarter-ton jungle cat into an air. So the interaction among all the motor proteins, as you might predict, like feature complementary arrangement of ionic, hydrogen bonding and hydrophobic groups at protein binding sites. So in this topic, we will discuss about the major protein of muscle and how that skeletal muscle protein translate chemical energy into the motion. So how it moves, right? So as we know, so the major protein of muscle is that we right, actin and myosin, right? So the contractile force of a muscle is generated by the interaction of these two proteins, myosin and actin. So these proteins are arranged in a filaments that undergo transient interaction. So what they undergo transient interaction with each other and may slide past each other to bring about contraction of the muscles. And together, actin and myosin make up more than 80% of the protein mass of the muscles, right? Right. So these two are the major proteins of muscles, right? And they almost made almost 80% of this two together of the mass of the muscles, right? All right. So the myosin. So first we'll discuss about the myosin. All right. So the myosin has a 6 subunits. So this protein has 6 subunits, right? From the six, two are heavy chain. So from this six, two are heavy chain and four light chain, right? The overall molecular mass of this is 520,000, right? But like it has 6 subunits, this protein has, and two heavy chains and four light chains. So the how the structure of this myosin is arranged. So I will draw a diagram here. So so the so what happens at the carboxy terminal, right? So the carboxyl terminal. So it's like I will try it. Something like this, and maybe something like this. So for example, this part is the tail, this is here head, all right, and this is the carboxy terminal, and this here is amino terminal, right? So in this structure, so at this carboxy terminal, so this is arranged like an extended alpha helices. So as we studied in alpha keratin, right? So that's the same here, coil as a alpha helix structure, right? Wrapped around each other, as you can see in a fibrous form, and this is a left-handed alpha helix, coiled coil similar to the alpha keratin, right? And at this amino terminus, so this each heavy chain, so there are two heavy chains, so these are the two heavy chains, has a large globular domain. So this has a globular domain, so that means this both can act as a separate entity and containing a site where the ATP is hydrolyzed. So on this site ATP can be hydrolyzed when it can be, so we'll see when how the muscles contract. So this on this side the ATP is stored and it is hydrolyzed at this side, and these two are the heavy chain, right? So this is the total heavy chain and this two globular domain, right? And the light chains, so the light chains are associated with this globular domain. So light chains is like this. So here is this. So maybe like this, something like this. Let's associate something like this. And the brown region you can see, so that might be the light chain associated with the globular domain. All right, so this is associated with the globular domain. So when the myosin is treated briefly, so when we will like split this myosin with the protease, with the enzyme that is trypsin, right? So splitting this with protease trypsin. All right. Right. So when we'll treat this myosin with this cleaving agent protease trypsin, so what it happens? So it will split. So this portion, for example, and something like this. All right. So this will split from here after treating with trypsin, right? And this will be, so this dividing component, protein components, will be called light myromyosin. So light myromyosin, and this will be heavy myromyosin, right? So light and heavy after splitting with the enzyme trypsin. So this will be splitted from here. So this just the alpha, this alpha helices structure like this will be light myromyosin and this will be heavy myromyosin, right? So and furthermore, so if we treat this part, this here part, again with the enzyme papain, so that's this enzyme papain, the same, will split this part. So what happens? So it will be separated like this, for example, and right, so this will be separated like this. So this is, this both one will be S1 and this will be just name and then S2. All right, so this globular domain, this is the myosin subfragment, that is the S1, or simply the myosin head group is liberated from the heavy myromyosin, right, by the splitting of the enzyme papain, leaving myosin subfragment S2, right, this is separate, and the S1 fragment is a motor domain that makes muscle contraction possible. So this segment is the domain that makes the muscle contraction possible. So this is how it is studied, the myosin, right? This is on the molecular level after splitting it, right? So in muscle cells, molecules of myosin aggregate to form the structure called thick filaments. So in the muscle cells, these molecules of this myosin aggregates to form the structure that is called a thick filament in the muscle. So like it, for example, this is like this here. So this will like keep aggregating, and it is a bipolar structure, so it is on the both side, right? So it will like aggregate and make a thick filament like this, whatever. Something like this. So this is a thick filament from the myosin, they aggregate and make the thick filament of the muscle cells. So this is the rod-like structure are the core of the contractile unit. So and within this thick filament, several hundred myosin molecules are arranged with their fibrous tails associated to to form a long bipolar structure, right? So it has both poles, right? And the globular domain project from either end of this structure in a regular stacked array. So it like this, right? So this was myosin, the structure of myosin. And the second major muscles protein, that is actin, is abundant in almost all the eukaryotic cells, right? So the second one is actin, right? So in muscles, so the molecules of this actin is a monomeric actin called G-actin, right? And associate this G-actin associate to form a long polymer called F-actin. All right, so this actin is a monomeric actin, right? Monomeric actin, and it's called G-actin. G-actin. So how it forms the thin filament? So this G-actin associate to form a long polymer called F-actin. So G-actin associate together to form F-actin. Right. So for example, this is a G-actin, a small one. So it's associate together like this. And it forms a thin filament like a small circle. So this associate together. So the thin filament consists of this F-actin along, and with this thin filament there is a different proteins, that is troponin and tropomyosin. So right, so I will write down here, right? So the thin actin filament. So thin filament consists of F-actin, right? F-actin with the proteins, along with the proteins. So what are the proteins? That is troponin and tropomyosin. So these are the two proteins that is together with this F-actin as well in the thin filament. So the filament is part of this thin filament. How it assembles? As the successive monomeric actin molecules add to one end. So it starts adding to each other, and it makes this F-actin thin filament, right? And on addition to that, each monomer binds ATP as well. Each monomer, so each actin this small actin binds with ATP, then hydrolyzes it to ADP, right? Adenosine diphosphate. So every actin molecule in filament is complex to ADP. But this ATP hydrolysis by actin functions only in the assembly of this filament. All right. It does not contribute directly to the energy expended in the muscle contraction. And each actin monomer in the thin filament can bind it tightly and specifically to one myosin head group, right? So this each actin monomer in this thin filament can bind tightly and specifically to one myosin head group. All right. So this was myosin, and this is the actins. So this is a thick filament, and this is the thin filament. All right. Right, so apart from this protein, there are other proteins in the muscle cells as well, in the small number though. So the skeletal muscles consist of parallel bundle of muscle fibers, right? Each fiber a single, very large multinucleated cell, 20 to 100 micrometer in diameter, right? So this skeletal muscles right? This is consist of parallel bundles of muscle fibers. parallel bundle of muscle fibers, right? And each fiber, a single one, each fiber, so it is very large and multinucleated, and the diameter is 20 to 100 micrometer in diameter, right? These are formed from many cells fused together, and a single fiber often span the length of a muscles as well. Each fibers contain 1000 myofibrils. Each fiber contains 1000 myofibrils, right? And its diameter is 2 micrometer in diameter, and each myofibril consists of vast number of regularly arrayed thick and thin filaments complex to other proteins as well. Right, so on your screen you can see the picture. I'm not drawing the picture, that's too complicated though, but on your screen you can see the picture. So this is how, so you can see the skeletal muscles, then the muscle fibrils, then the myofibrils, right? And then this myofibrils, there is a system of flat membranous vesicles that are called sarcoplasmic reticulums that surround each myofibril, right? So what that vesicle is called, that is called sarcoplasmic reticulum, right? So okay, I'll write it down for you that. So that is a system of a flat membranous vesicle, and that is called sarcoplasmic reticulum, right? Right, and this surrounds each myofibrils. So we'll write it down here, surrounds each myofibrils. All right. So when this is examined under the electron microscope, so muscle fibers reveal alternating region of high and low electron density called A bands and I bands, as you can see on your screen with the picture. So there it will show A bands and I bands, as you can see on your screen. So the A and I bands arise from the arrangement of this thick and the thin filaments, which are aligned and partially overlapping. So they are aligned together and maybe partially overlapping as well. So the I band is region of a bundle that in cross section would contain only thin filaments. So I band just contain thin filaments. So I will write it down here. I bands contains thin filament. On cross section they filament. All right, I band contains just the thin filaments on the cross section, and the darker A band stretches the length of the thick filaments and includes the region where the parallel thick and the thin filaments overlap as well. So the A band has thick filament, right? And also parallel, well, thick and thin filament as well when they overlap each other. Filament on the cross section, right? So while bisecting, the I band is a thin structure called the Z disc, right? So as you can see on your screen, so the I band is bisected. It's not dissected, it's bisecting, it's just dividing. Bisecting the I band is a thin structure that is called a Z disc, right? Bisecting I band, a thin structure that is called Z disc, right? So for example, this is the thin filament. So the middle part, like for example this is, that's bisecting the I band. So that thin structure is Z disc, right, perpendicular to the thin filament and serving, this serve as an anchor to which the thin filaments are attached, right? So this Z disc is like an anchor, so where these thin filaments are attached, right? And the A band too is also bisected. So bisecting A band. So when the, so what is bisecting a waveband? That is also a thin line. That line is called thin line called M line, or also, or you can see M disc as well, or right, Z disc or M disc. So this M region is a region of a high electron density in the middle of a thick filament. Right? For example, this is a thick filament and the bisecting region is the M disc. So this region is a high electron density in the middle of the thick filament, and the entire contractile unit consisting of bundles of thick filament interleaved at either end with the bundle of thin filament is called sarcomere. Right? So I'll write it down. So what is called a sarcomere? So the entire contractile unit consisting of bundles of thick and bundles of thick filaments, right? Interleaved at either end. At either end with the bundles of thin filaments, right? Filament is called the sarcomere. Yeah. Right? Right, so this is called sarcomere, and why this is arranged like this? So the arrangement of this interleaved bundles allow the thick and the thin filaments to slide past each other, causing a progressive shortening of each sarcomere. Right? And these thin actin filaments are attached at one end, right, to the Z disc in a regular pattern. The assembly includes the minor muscles as well, proteins. So that is alpha-actinin, dasmin, and vimentin as well. So beside this protein, there is alpha-actinin, dasmin, and vimentin. So I'll write it down here with this. Alpha-actinin, dasmin, and vimentin. So these are the other proteins as well. Right? Thin filaments also contain a large protein as well. But these are the small protein. The large protein that is called nebulin as well. All right. Large protein that is nebulin. All right. So the nebulin structure is like almost 7,000 amino acid residues, and this is thought to be the structure of an alpha helix that is long enough to span the length of the entire filament, right? And the M line, the same as the Z line that we discussed, M line similarly organize the thick filaments, right? It contains protein. The M line contains paramyosin, C protein, and M protein as well, right? So these are for the this actin thin filament, right? So for example, I'll write it down here for this. So this thick filament contain paramyosin, C protein, and also protein. All right, and there is another class of protein as well that is called titins. That's a large protein as well. Protein. Titins, right? This is the largest single polypeptide chain discovered far. This is almost like 26,926 amino acid residues chain. And the titin, and that titin is of the human cardiac muscle. That is 26,926. This link the thick filament to the this titin link the thick filament to the Z disc, providing additional organization to overall structure. And among their structural function, the protein nebulin and the titin are believed to act as a molecular rulers, regulating the length of the thick and thin and the thick filament respectively. Right? And the titin extend from Z disc to the M line, regulating the length of the sarcomere itself and preventing overextension of the muscles. Right? So the titin function is as well to regulate the length of the sarcomere itself and preventing overextension of the muscle. Right, so that is something important. So I'll write it down. Right, so I won't remove this anyway. Right, so the function of nebulin, titin, right, is like a molecular ruler to regulate the length of regulating the length of the actin and myosin. The length of thin and thick filaments, right? The titin as well, the function of titin is that extend from Z disc to M line, regulating the length of the sarcomere as well, right? Titin, this extends from the Z disc to M line, that will regulating the length of sarcomere, right, itself, and preventing what it does, it prevents and preventing overextension of the muscles. Extension of muscles, right? Right, and the characteristics of the sarcomere length actually varies from one muscle tissue to the next in vertebrate. Why? Because largely due to the different variants of this titin in the tissues. All right, so this is the overall structure of the muscle. So the myosin, actin, and the few other proteins, that how it builds the muscles. Alright, thank you guys for watching, and don't forget to subscribe to the channel. We'll see you in the next video, and we'll discuss how the actin and myosin help in the muscle contraction. So that will be much more interesting, how your muscle contracts. Alright, thank you guys for watching. See you in the next video.